Chapter 14: Monounsaturated Fats and Adaptive Fluidity. Monounsaturated fats introduce controlled flexibility into membrane structure by inserting a single double bond into the fatty acid chain, creating a bend that disrupts tight packing without collapsing structural order. This geometric change prevents lipids from aligning as densely as saturated fatty acids, increasing lateral movement within the bilayer while still maintaining cohesion. The result is a membrane that can adjust its physical state in response to changing conditions without losing containment. This capacity for adjustment defines adaptive fluidity, where the membrane remains stable but capable of shifting its behavior as functional demands change. Fluidity determines how quickly and efficiently components within the membrane can move. Receptors must cluster, channels must transition between states, and vesicles must fuse and separate from the bilayer. These processes require space and mobility within the lipid environment. Monounsaturated fatty acids provide this space by introducing slight irregularity into packing, allowing lipids to slide past one another without creating large gaps. This supports dynamic processes while preserving the continuity of the membrane, preventing the increase in permeability that would occur if structural order were lost entirely. The balance between saturated and monounsaturated fats establishes a functional range of membrane behavior. Saturated lipids provide resistance to deformation and limit permeability, while monounsaturated lipids allow controlled movement and adaptation. Together, they create a membrane that can maintain gradients while still supporting dynamic processes. If monounsaturated content is too low, the membrane becomes overly rigid, restricting protein function and slowing processes that depend on movement. If too high, the membrane becomes less ordered, increasing permeability and reducing the ability to maintain controlled conditions. Monounsaturated fats also influence how membranes respond to temperature. As thermal energy increases, the presence of kinks in fatty acid chains prevents excessive ordering that would otherwise restrict movement at lower temperatures, while still maintaining enough cohesion to prevent destabilization at higher temperatures. This creates a buffer against temperature-driven changes in membrane behavior, allowing function to remain consistent across a broader range of conditions. Cholesterol interacts with monounsaturated fatty acids to refine this adaptive state. It positions itself between lipid molecules, reducing excessive movement when fluidity increases and preventing the membrane from becoming too rigid when fluidity decreases. In the presence of monounsaturated lipids, cholesterol helps maintain a controlled midpoint where the membrane can respond to changes without crossing into instability. This interaction allows the system to fine-tune its physical properties based on current demands. Adaptive fluidity directly affects signaling. Receptor activation often depends on the ability to cluster within specific regions of the membrane, and this clustering requires mobility within the bilayer. Monounsaturated fats support this process by enabling lateral movement while maintaining the structural integrity needed for signal localization. This ensures that signals can be initiated and propagated without interference from excessive rigidity or disorder. Energy systems also depend on this balance. Transport processes and gradient maintenance require membranes that can adjust without leaking. Monounsaturated fats allow minor structural shifts that accommodate changes in activity while preserving containment. This reduces the energy required to maintain conditions, as the membrane can adapt rather than resist all change or collapse under pressure. Incorporation of monounsaturated fats occurs through the same remodeling cycles that govern overall membrane composition. Over time, their presence adjusts the functional properties of the membrane, shaping how it responds to mechanical, thermal, and signaling inputs. This gradual integration ensures that adaptive fluidity reflects sustained input rather than transient fluctuations. Monounsaturated fats therefore provide the capacity for membranes to remain responsive without sacrificing stability. By introducing controlled disruption into lipid packing, they enable movement, support dynamic processes, and allow the membrane to adjust to changing conditions while maintaining its role as a boundary that enforces separation and control across the system. Controlled flexibility in membranes. Controlled flexibility arises from the ability of the membrane to permit movement without losing structural continuity. The bilayer must allow lipids and proteins to shift position, change conformation, and reorganize locally while still maintaining a continuous barrier that preserves gradients and prevents uncontrolled exchange. This is achieved through the integration of monounsaturated fatty acids, whose single double bond introduces a bend in the hydrocarbon chain. This bend disrupts perfect alignment between neighboring lipids, creating space for lateral movement without generating the large defects that increase permeability. This flexibility operates within defined limits. Lipids must move enough to support processes such as receptor clustering, vesicle fusion, and channel gating, but not so freely that the membrane becomes disordered. Monounsaturated fats provide this intermediate state by reducing packing density just enough to allow movement while maintaining sufficient cohesion to hold the bilayer intact. The membrane can deform locally to accommodate functional demands, then return to its baseline state once the demand subsides. Protein function depends on this controlled environment. Embedded proteins rely on the surrounding lipid matrix to provide both support and mobility. If the membrane is too rigid, conformational changes required for activity are restricted, slowing signaling and transport. If too fluid, the lack of structural constraint alters how proteins shift between states, reducing precision. Controlled flexibility ensures that proteins operate within a consistent mechanical context, allowing predictable transitions and coordinated activity. Flexibility also determines how membranes handle localized stress. During processes such as endocytosis or exocytosis, regions of a membrane must curve and reshape without fracturing. Monounsaturated lipids facilitate this by allowing lipids to rearrange under force, distributing strain across the bilayer. This prevents the formation of discontinuities while enabling the membrane to change shape as required. The interaction with cholesterol further refines this flexibility. Cholesterol inserts between lipid molecules and adjusts how much movement is permitted. In areas where fluidity increases, it restricts excessive motion, maintaining cohesion. In areas where rigidity dominates, it disrupts tight packing enough to allow movement. This modulation keeps flexibility within a functional range, preventing extremes that would impair membrane behavior. Electrical and transport functions are also influenced by this property. Ion channels and transporters depend on a stable yet adaptable membrane to maintain gradients while allowing controlled passage of substrates. Controlled flexibility ensures that these proteins can open, close, and cycle efficiently without introducing unintended leakage across the bilayer. Over time, remodeling cycles adjust the level of flexibility by altering the proportion of monounsaturated lipids within the membrane. This gradual process aligns membrane behavior with sustained input and functional demand rather than short-term fluctuations. The result is a membrane that reflects its environment while maintaining stability. Controlled flexibility therefore defines how the membrane accommodates movement without compromising its role as a boundary. By allowing localized rearrangement while preserving overall structure, the bilayer supports dynamic processes and maintains the conditions required for signaling, transport, and energy production because the system can adjust without losing the containment that underlies all function. Signal modulation through fluid balance. Signal modulation begins with how freely components within the membrane can move and organize in response to input. Receptors do not operate in isolation. They depend on lateral movement within the bilayer to cluster, separate, and interact with downstream partners. Fluid balance determines how quickly and precisely these arrangements form. When the membrane maintains controlled fluidity, receptors can assemble into functional groups at the right time and location, then disperse once signaling is complete. This movement is constrained enough to prevent random aggregation, but permissive enough to allow rapid reorganization in response to changing conditions. Fluid balance influences the threshold required for signal initiation. In a membrane with appropriate mobility, receptors encounter one another with predictable frequency, allowing activation to occur only when sufficient input is present. If the membrane becomes too rigid, these encounters are limited, raising the threshold and slowing signal initiation. If too fluid, encounters become excessive and unstructured, lowering the threshold and increasing the likelihood of spontaneous or amplified signaling. The membrane therefore sets the baseline sensitivity of the system by controlling how often signaling components come into contact. Signal amplification depends on the ability to concentrate activity within specific regions. Microdomains form when lipids and proteins co-localize, creating areas where signaling molecules are enriched. Controlled fluidity allows these domains to assemble without dispersing prematurely. Monounsaturated lipids enable the lateral movement required for this assembly, while surrounding structural elements prevent the domain from dissolving into the broader membrane. This balance allows signals to be strengthened locally without spreading indiscriminately across the cell. Termination of signaling also relies on fluid balance. Once a signal has been transmitted, components must separate and return to a baseline state. If the membrane is too rigid, this separation is delayed, prolonging signaling beyond its intended duration. If too fluid, components may never remain associated long enough to complete the signal. Controlled fluidity allows both assembly and disassembly to occur with appropriate timing, ensuring that signals begin and end within defined windows. Ion channel behavior is similarly affected. Channels depend on the surrounding lipid environment to maintain the structural conditions required for opening and closing. Fluid balance determines how easily these conformational changes occur and how stable each state remains. Excess rigidity restricts transitions, while excess fluidity destabilizes them. The membrane sets the conditions under which electrical signals are generated and propagated by controlling this balance. Signal fidelity depends on consistency across the membrane. Variations in fluidity create regions with different signaling properties, introducing noise into the system. Controlled balance reduces these variations, ensuring that similar inputs produce similar outputs regardless of location. This uniformity allows coordinated responses across the cell, rather than fragmented or conflicting signals. Metabolic state feeds into this system through remodeling cycles that adjust lipid composition over time. The proportion of monounsaturated lipids influences how fluid the membrane becomes, linking sustained input to signaling behavior. As composition shifts, the baseline sensitivity, amplification, and termination of signals shift with it, altering how the cell interprets and responds to its environment. Signal modulation through fluid balance, therefore, reflects the physical state of the membrane rather than isolated biochemical pathways. By controlling movement within the bilayer, the membrane defines how signals are initiated, amplified, and resolved. When this balance is maintained, signaling remains precise and coordinated. When it drifts, the system becomes either unresponsive or excessively reactive because the structural conditions that govern interaction and timing are no longer aligned with functional demand. Integration with saturated structures. Monounsaturated fats do not operate as independent modifiers of membrane behavior. Their function emerges only within the structural framework established by saturated lipids. Saturated fatty acids define baseline packing density, membrane thickness, and resistance to deformation. Monounsaturated fatty acids integrate into this framework by introducing localized disruption that allows movement without dissolving the overall structure. The relationship is not additive. It is interdependent. Without a stable saturated backbone, the flexibility introduced by monounsaturated lipids leads to loss of cohesion. Without monounsaturated input, the saturated framework becomes restrictive, limiting dynamic processes required for signaling and transport. Integration occurs at the level of molecular packing. Saturated chains align tightly, forming regions of high order. Monounsaturated chains insert between these aligned lipids, creating controlled spacing that permits lateral movement. This spacing is not random. It is constrained by the surrounding saturated matrix, which prevents excessive displacement. The result is a membrane that maintains continuity while allowing localized rearrangement. Movement occurs within boundaries defined by structural density, preserving containment while enabling function. Cholesterol stabilizes this integration by occupying the spaces created between lipid molecules and regulating how much movement is allowed. In regions where saturated lipids dominate, cholesterol prevents excessive rigidity by interrupting overly tight packing. In regions where monounsaturated lipids increase fluidity, it limits excessive motion by reinforcing interactions between neighboring molecules. This creates a dynamic equilibrium in which flexibility is continuously adjusted but never exceeds the limits required for structural integrity. Protein function reflects this integrated state. Receptors and channels require both a stable platform and the ability to undergo conformational change. The saturated framework provides the mechanical support that maintains protein orientation and spacing. Monounsaturated lipids allow the surrounding environment to shift as proteins change shape during activation. Without this integration, proteins either become constrained within an inflexible matrix or lose the structural context required for precise operation. Mechanical stress is distributed through this combined structure. Saturated lipids absorb and distribute force across the membrane, while monounsaturated lipids allow localized deformation that prevents fracture. This combination reduces the likelihood of structural failure under load. The membrane bends, stretches, and recovers without losing continuity because flexibility is embedded within a stable framework. Electrical properties are also governed by this integration. The saturated backbone maintains low permeability, preserving ion gradients and electrical potential. Monounsaturated lipids allow the subtle adjustments required for channel activity and membrane deformation without introducing leakage. Together, they maintain the balance between insulation and responsiveness that defines electrical stability. Remodeling cycles continuously adjust the ratio of saturated to monounsaturated lipids, refining this integration over time. Changes in input alter how these components are distributed within the membrane, shifting the balance between rigidity and flexibility. This process aligns membrane behavior with sustained conditions rather than transient fluctuations, ensuring that structure reflects ongoing demand. Integration with saturated structures therefore defines how membranes achieve both stability and adaptability. Monounsaturated fats provide movement, but only within the constraints imposed by saturated lipids. This relationship allows the membrane to function as a controlled environment where dynamic processes occur without compromising the boundary that supports them, because flexibility is embedded within structure rather than existing in opposition to it. Functional tuning of cellular environments. Cellular function is not set by individual pathways acting in isolation. It is tuned through the physical state of the membrane, which defines how signals are received, how substrates move, and how energy is maintained. The ratio and distribution of saturated and monounsaturated lipids establish this state by determining packing density, lateral mobility, and permeability. These properties do not operate as abstract characteristics. They directly shape the behavior of proteins, gradients, and signaling networks embedded within the bilayer, creating an environment that either supports precise function or introduces variability. Tuning begins with how the membrane sets baseline conditions. A structure with sufficient saturation maintains low permeability and stable gradients, providing a consistent internal environment. Monounsaturated lipids introduce controlled mobility, allowing the system to adjust without disrupting this baseline. The combination determines how sensitive the cell is to input. Increased rigidity raises the threshold for activation by limiting movement and interaction between signaling components. Increased fluidity lowers that threshold by allowing more frequent encounters. The membrane therefore defines whether the cell responds selectively or reacts to minimal input. Receptor behavior is directly shaped by this environment. Activation depends on the ability to cluster and interact with downstream proteins. Controlled fluidity allows these clusters to form when needed, while structural density prevents uncontrolled aggregation. This balance determines signal strength and duration. A membrane that maintains proper tuning supports discrete, time-limited signaling events. When tuning is lost, signals either fail to initiate or persist beyond their intended duration, altering downstream processes. Transport processes are similarly affected. Nutrient uptake, ion exchange, and waste removal all depend on proteins that operate within the membrane. These proteins require both structural support and the ability to transition between states. Saturated lipids maintain the conditions necessary for selective permeability, while monounsaturated lipids allow the conformational changes required for transport. The efficiency of these processes reflects how well the membrane maintains this balance, as excessive rigidity or fluidity disrupts the coordination required for controlled exchange. Energy systems operate within the same constraints. Mitochondrial membranes rely on tight control of proton gradients, and any increase in permeability reduces efficiency. Structural density preserves these gradients, while controlled flexibility allows the membrane to adapt to changes in energy demand without introducing leakage. The ability to maintain output under varying conditions depends on how well the membrane sustains this tuned state. Local variations within the membrane create specialized environments that support specific functions. Microdomains form where lipid composition and protein content differ from the surrounding bilayer, concentrating signaling or transport activity. Controlled tuning allows these domains to exist without destabilizing the broader membrane. Movement between domains remains regulated, ensuring that localized activity does not spread in an uncontrolled manner. Remodeling cycles adjust this tuning over time. As lipids are replaced, the balance between saturated and monounsaturated components shifts, altering membrane behavior in response to sustained input. This process ensures that the cellular environment reflects long-term conditions rather than short-term fluctuations, aligning structure with functional demand. Functional tuning of cellular environments therefore emerges from the integrated properties of the membrane. By controlling rigidity, mobility, and permeability, the bilayer defines how signals are processed, how materials are exchanged, and how energy is maintained. When this tuning is maintained, cellular processes operate with consistency and precision. When it drifts, variability increases, and the system begins to produce outputs that no longer match input because the structural conditions that govern behavior are no longer aligned with the demands placed upon them.